Good day, this is Professor Will, CBMD PhD. Today is Monday, July 8th, 2019. It is 7:51, 50 seconds Eastern Standard Time. This is a continuation of my pathophysiology. So, let's hop to it here. Imprinted spears fight breast cancer. Inhibition of HER2 on tumor cells by molecularly imprinted nanoparticles. Date July 3rd, 2019. Source Wiley. Summary. A particularly aggressive metastasizing form of cancer, HER2-positive breast cancer, may be treated with nanoscopic particles imprinted with specific binding sites for the receptor molecule HER2. The selective binding of the nanoparticles of HER2 significantly inhibits multiplication of the tumor cells. Breast cancer is the most common form of cancer in women and one of the leading causes of death. About 20 to 30% of breast cancer cases involve the very poorly treatable HER2 positive variety. HER2 stands for Human Epidermal Growth Factor Receptor 2, a protein that recognizes and binds to a specific growth factor. HER2 spans across the cell membrane. One part protrudes into the interior of the cell, the other is on the cell surface. As soon as the growth factor docks, the extracellular parts of HER2 bind into a heterodimer with a second closely related HER, such as HER1 and HER3. This triggers a multi-step signal cascade within the cell, which is critically involved in processes like cell division, metastasis, and the formation of blood vessels that supply the tumor. HER2 positive tumor cells contain significantly higher concentrations of HER2. One current therapy for early stage HER2 positive tumors is based on binding an antibody to HER2 block the dimerization. Researchers have now developed molecularly imprinted biocompatible polymer nanoparticles that recognize HER2 just as specifically as an antibody in order to prevent the dimerization. Nanoparticles can be molecularly imprinted in that, to simplify, a polymerizable mixture is polymerized into nanospheres. In the presence of the big molecules they are supposed to recognize later. The biomolecules act as a kind of stamp, leaving nanoscopic imprints in the sphere. These then perfectly fit the molecules they were imprinted with and bind to them specifically. In contrast to antibodies, the nanospheres are easy and inexpensive to produce and are chemically stable. For the imprinting process, the researchers use a special method, boronate, affinity controllable oriented surface imprinting that is particularly controllable and makes it possible to imprint using chains of sugar building blocks, glycans as templates. Many proteins contain specific sugar chains. These are unique, like a protein fingerprint. The researchers used this kind of glycan from the extracellular end of the HER2 proteins as their stamp. This allowed them to produce imprinted nanoparticles that specifically recognize HER2 and selectively bind to it, inhibiting the dimerization. They were thus able to significantly reduce the multiplication of tumor cells in vitro and the growth of tumors in mice. In contrast, healthy cells were essentially unaffected. Good day, this is Professor Will, CBMD PhD. Today is Monday, July 8, 2019. It is 8:19, 18 seconds Eastern Standard Time. And this is a continuation of my scientific research. So let's hop to it here. Artificial DNA can control release of active ingredients from drugs. Date June 28, 2019. Source Technical University of Munich, TUM. Summary: a drug with 3 active ingredients that are released in sequence at specific times. What was once a pharmacologist's dream is now much closer to reality. With a combination of hydrogels and artificial DNA, nanoparticles can be released in sequence under conditions similar to those in the human body. It is becoming much more common for patients to be treated with several different medications. It is often necessary for the patient to take them at fixed intervals, a limitation that makes everyday life difficult and increases the risk of doses being skipped or forgotten. Researchers have now developed a process that could serve as the basis for medication containing several active ingredients that would reliably release them in the body in a predefined sequence at specified times. For example, an ointment applied to surgical incision could release pain medications first. Followed by an anti-inflammatory drug, and then a drug to reduce swelling. One active ingredient after the other. Ointments or creams releasing their active ingredient with a time delay are not new in themselves. With the drugs currently in use, however, there is no guarantee that 2 or more active ingredients will not be released into the organism simultaneously. To test the principle behind their idea, researchers used nanoparticle-sized silver iron oxide and gold particles embedded in a special gel-like substance known as a hydrogel. They then used a spectroscopic method to track the exit of the particles from the gel. The particles selected by the researchers have similar motion characteristics within the gel to the particles used to transport real active ingredients, but are easier and cheaper to make. A special ingredient controlling the nanoparticles in artificial DNA. In nature, DNA is above all the carrier of genetic information. However, researchers are increasingly exploiting another property, the ability of DNA fragments to be combined with great accuracy, both in terms of the types of bonds and their strength. For example, to build machines on the nanometer scale. The DNA cascade compressed and then released at the right instant. The silver particles were released first. In the initial state, the particles were bound together by DNA fragments designed by special software. The resulting particle clusters are so large that they are unable to move in the hydrogel. However, when a saline solution is added, they separate from the DNA. They can now move in the gel and drift to the surface. Because the saline solution has approximately the same salinity as the human body, we were able to simulate conditions where the active ingredients would not be released until the medication is applied. The mesh-like DNA structure surrounding the iron oxide particles consists of two types of DNA. The first has one end attached to the iron oxide particles. The second is attached to the loose ends of the first type. The second type is attached to the loose ends of the first type. These structures are not affected by the saline solution. The iron oxide particles can only be released when the first clusters have dissolved. This event releases not only the silver nanoparticles, but also DNA, which eliminates the connection DNA of the second cluster without forming connections itself. This releases DNA fragments, which in turn act as the key to third DNA nanoparticles combination. The consistency of ointments makes them the most obvious solution for a hydrogel-based approach. However, this principle also has the potential to be used in tablets that could release several effective ingredients in the body in a specific order. Good day, this is Professor Will, CBMD, PhD. Today's Monday, July 8, 2019. It is 8:41:28 seconds Eastern Standard Time. This is a continuation of my scientific research on pathophysiology, so let's get started here. First observation of native ferroelectric metal, date July 6, 2019. Source, ARC Central of Excellence of Future Low Energy Electronics Technologies. Summary. Researchers describe the first observation of native ferroelectric metal, a native metal with biostable, electrically switchable spontaneous polarization states. The hallmark of ferroelectricity, the study found coexistence of native metallicity and ferroelectricity of bulk crystalline tungsten detelluride, WTE2, at room temperature. A van der Waals material that is both metallic and ferroelectric in its bulk crystalline form at room temperature has potential for nanoelectronics application. Ferroelectric background. Ferroelectricity can be considered an analogy to ferromagnetism. Ferromagnetic material displays permanent magnetism, and in layperson's terms is simply a magnet with north and south poles. Ferroelectric material likewise displays an analogous electrical property called a permanent electric polarization, which originates from electric dipoles consisting of equal but oppositely charged ends or poles. In ferroelectric materials, these electric dipoles exist at a unit cell level and give rise to a non-vanishing permanent electric dipole moment. This spontaneous electric dipole moment can be repeatedly transitioned between two or more equivalent states or directions upon application of an external electric field, a property utilized in numerous ferroelectric technologies. For example, nanoelectronic computer memory RFID cards, medical ultrasound transducers, infrared cameras, submarine sonar, vibration and pressure sensors, and precision actuators. Conventionally, ferroelectricity has been observed in materials that are insulating and semiconducting rather than metallic, because conduction electrons in metals screen out the static internal fields arising from the dipole moment. Bulk single crystalline tungsten detelluride, WTE power 2, which belongs to a class of materials known as transition metal dichylkygenides, TMDCs, was probed by spectroscopic electron transport measurements, conductive atomic force microscopy, CAFM, to confirm its metallic behavior, and by piezo response force microscopy, PFM, to map the polarization detecting lattice deformation due to an applied electric field. Ferroelectric domains, i.e. the regions the oppositely oriented direction of polarization, were directly visualized in freshly cleaved WTE power 2 single crystal. Spectroscopic PFM measurements with top electrode and capacitor geometry was used to demonstrate switching of the ferroelectric polarization. Switchable electric dipole moment of ferroelectric materials could, for example, be used as a gate for the underlying 2D electron system and artificial topological insulation. In comparison with conventional semiconductors, the very close sub-nanometer proximity of the ferroelectric electron dipole moment to the electron gas and the atomic crystal ensures more effective switching, overcoming limitations in conventional semiconductors where the conducting channel is buried tens of nanometers below the surface. Topological materials are investigated within Fleet's research team-wide, which seeks to establish ultra-low resistance electronic paths with which to create a new generation of ultra-low energy electronics. Good day, this is Professor Will, CBMD PhD. Today is Monday, July 8, 2019. It's 9:07:11 seconds Eastern Standard Time. And this is the end of this module on pathophysiological research. Hope you enjoyed listening. Have a great day.